What I will talk about is a paradigm shift in alloimmunity, especially humoral alloimmunity, where we now understand the molecular mechanisms much better, and this will provide novel risk stratification options that we can talk about. This is again the collaborative transplant study from Gerald Opels in Heidelberg. That's one of the largest transplant registries, and this nicely shows that alloimmune responses, defined here by HLA antigen level mismatch, are associated with adverse long-term graft outcome. We also see that the difference between the different HLA mismatch groups is predominantly in the first year after transplantation, and afterwards the slopes are almost parallel. When we look about the change in graft survival over the last decades, this is a work from Thomas Weckele from our institution, that nicely shows that acute rejection rates in the first year, that's the blue line, significantly decreased over the last 20 years with the introduction of highly efficient, especially focused on T-cell activation immunosuppression. But long-term graft survival remained more or less stable at the rate of 4% for every year after the first year, and this remains stable over the last 20 years. This is probably the main work on this concept of humoral alloimmunity. That's from Phil Halloran's group in Edmonton, that shows that after the first year, the predominant finding in histology is antibody-mediated rejection. And this is driving the long-term graft attrition after kidney transplantation. So we see TCMR, the red line, earlier after transplantation, and this completely fades, and then we only see antibody-mediated rejection. So this kind of shaped our current antibody-focused view of alloimmunity in the long run. This is just a basic slide that shows how this indirect allorecognition, that's some sort of conventional immune response, drives the antibody formation. Antigens are bound by B cells with the receptor that binds the antigens, and they are internalized, presented to T cells that are needed for activation, and this T cell goes to the bone marrow and becomes a plasma cell, secreting antibodies that are detecting the antigen in the graft and cause graft vasculopathy, or remain as memory B cells. So this is our current understanding of the allo-antibody development. When we now look at what the antibody is actually binding, that is, we could identify specific epitopes within the molecular structure of the HLA epitope. And the antibody specifically binds polymorphic residues on the cell surface. And there have been different ways to describe these polymorphisms or these non-self residues. There's the applet concept, then there's a simple amino acid exchange concept, and there was a triplet concept that only looked at 3 adjacent amino acids, or there are physical chemical properties of the molecule that render it different from the outside and that give way to antibody epitopes. So we now understand that the HLA molecule consists of different epitopes, for example for donor 1 or donor 2, that can be mismatched at different positions within the molecule when you transplant this organ into the recipient. For example, donor 2 has 3 of those non-self epitopes and donor 2 only has 2. So the likelihood that donor 2 will develop a detrimental alloimmune response is considered to be lower than for donor 1, who has more non-self epitopes present on the transplanted organ. And this is a work by Chris Weber. He's from Manitoba in Canada, from Peter Nickerson's group, and they're leading the field in this epitope mismatch concept. And they just looked at HLA-DR serotype mismatch 1 or 2, or DQ mismatch 1 or 2, and they accounted for the different epitopes that this represented. So an individual with an HLA-DR mismatch of 2 can have only 5 mismatched epitopes, whereas an individual with an HLA-DR mismatch of 1 can have up to 25 of those different epitopes. And this shows that the actual risk of an alloimmune response may be higher despite the lower HLA serotype mismatch level. And this was the largest study to date in 2,000 individuals from Berlin by Nils Lachmann. They showed that if you stratify transplants, they did a retrospective analysis of their HLA database, by the number of epitope mismatches, they could see that the more epitope mismatches you had, the worse your graft survival was. And we in our cohort, we then specifically looked at individuals with an A mismatch level of 3, And we could further separate risk that can be attributed to the epitope level within this HLA serotype mismatch level group. So this epitope mismatch gives us additional information about the risk for a detrimental alloimmune response that can be translated into long-term graft outcome. Again, back to Peter Nickerson and Chris Wiebe, who made a retrospective analysis of their cohort of 286 patients, and they specifically looked at HLA class 2 mismatch. And they could identify a number of mismatched epitopes for the DER locus at 0.0 that virtually carried no risk, or up to 16 on the DQ level, where the risk was virtually 0 to develop donor-specific antibodies. But if you had more mismatched epitopes with your donor, the risk to develop an antibody significantly increased over time. And this is probably why this is going to change how we treat our patients. This is a slide also from their group that showed that only patients with a high epitope mismatch at the DR locus who were not adherent to immunosuppression developed antibodies. And those patients who were non-adherent, they were not taking their TAC or their MPA, and despite having, and they only had a low mismatch, did not develop antibodies. So we might identify individuals who are at an increased risk of rejection when they're not compliant with their medication, or we could identify individuals having a high risk, where we might, having a low risk, where we might be able to reduce our immunosuppression because they were non-adherent and had no mismatch and had low mismatch level and showed no development of antibodies. So this is how this may change the field over next year, and there's prospective trials now underway in Europe. And in Canada and the USA, because it was approved to be a surrogate biomarker for graft outcome by the FDA recently. And our own work now tried to evolve this concept, as was nicely laid out by the previous speaker, to non-HLA polymorphic cell surface structures. And this is again a quote from this Oppel's work from the CTS in an HLA-matched siblings, and they specifically looked at patients who had positive panel-reactive antibody results despite having no HLA mismatch. And those antibodies were supposed to be directed against non-HLA cell surface structures. And you can see that in the first year there was no difference in graft survival, but for the long-term graft survival up to 10 years, those who were sensitized against non-HLA antigens showed a significantly reduced graft survival. So this kind of showed that there is more to alloimmunity than HLA in terms of antibodies. And this is again the concept of the endothelial cell surface, the endothelial cell antibodies that include all the before mentioned antibodies, and I won't cover this in more detail, but the cell surface contains multiple proteins that might serve as potential antigenic structures in the alloimmune response. So over the last 10 years, two great international consortia, the 1000 Genomes and the HapMap project, they sequenced several hundreds of individuals, and they uncovered that each individual differs by other individuals by up to 10,000 so-called non-synonymous single nucleotide variants. These are genetic variants that change the amino acid structure of proteins and thereby have polymorphic residues on cell, potentially also on cell surface proteins. So this was kind of the ground laying work that kind of showed us that there's polymorphisms outside the HLA region that might play a role in alloimmunity. So we hypothesized that if we have polymorphic and mismatched donor and recipient pairs, and we specifically focus on transmembrane and secretin proteins, when we transplant an organ with an altered cell surface structure by a changed amino acid, this will cause an alloimmune response in the recipient. We sequenced 500 donor and recipient pairs, and we identified up to 60,000 mismatched variants for this cohort. We excluded all variants in the HLA region, so specifically focus on the impact of non-HLA polymorphisms. And then we aligned the genomes of donors and recipients and identified all those mismatched variants for each pair, and we quantified it. In this cohort of 477 donor and recipient pairs, we only looked at first kidney transplant deceased donors with stable graft function at 3 months to rule out most important influential factors. We identified a median of almost 1,900 of these mismatched cell surface proteins. And when we plotted it in the quartiles of mismatch, we could see that those that had the least mismatch had the best graft survival compared to those individuals with a higher level of mismatch. This transmembrane and secreted protein mismatch was significantly associated with graft outcome, with graft survival, also when we adjusted for the HLA epitope mismatch as described before. So this shows that there is an alloimmune response against the graft outside the HLA that is associated with graft survival. To prove an alloimmune response, we designed overlapping peptide probes where we incorporated 16-mer peptides with the amino acid mismatch at position 8 and 12. And we also used the wild-type version or the self version. And then we looked for mismatch-specific antibody response by incubating serum from recipients at specified time points after transplantation. And this is just an example. For example, for the interleukin-1 receptor-like 1 protein, we could identify in one individual who was mismatched at time-dependent, at time of rejection, and mismatch-specific antibody response, while the other individuals were not mismatched for these variants and did not show an antibody response. And the same is true for this other protein. In total, we identified 16 of those mismatched peptides where we could identify a directed alloimmune response. So, summarizing, what we saw is that each individual that each transplant pair carries its own pattern of incompatibilities. It's unique for each pair, and the more mismatches you have, the worse your graft survival was, and we could identify antibodies against those mismatched peptides. So this can also be taken further because there is additional genetic variants that result in the complete loss of a gene product. These are called loss of function variants. And there has been a lot of scientific attention put to them because especially the PCSK9 inhibitors were identified as a complete loss of function variant in a small population of African Americans who did not show any risk for cardiovascular disease despite having a severe number of risk factors. So loss of function variants are present and common in humans. They most of the time do not have any effect on the phenotype, but some of them can be druggable targets. And in our specific concept, we hypothesized that when a recipient carries a homozygous loss of function variant, so he's completely absent of this specific gene product, when the donor introduces this protein through transplantation, this will also induce an alloimmune response. A very prominent example for this concept is the anti-GBM antibody disease following transplantation Alport syndrome. That's described in the literature occurring about 5%. We looked at our center data and we didn't find any case, but it's described in the literature that when you have a colic gene loss of function variant and you receive a kidney, you can develop antibodies against the glomerular basement membrane similar to Goodpasture syndrome. So this is kind of a proof of principle that this can cause adverse outcome following transplantation. And then another group from the US, from Columbia, Steers et al., used a similar approach, and they looked at copy number variants that were associated with deletions of specific genes. They started with a high number of genes, and then they, after quality control and allele frequency, they came up with a candidate list of 44 deletions, and they tested for association with acute rejection, and they could identify a single variant that tagged the loss of gene product for a specific gene, in this case for LIMS1, and that was associated with acute rejection. And they validated this finding in a multi-center cohort in Europe and the US, and they could show that those individuals who carrying a homozygous copy number loss of LIMS1 who received the transplant with a functioning copy showed an increased rate of graft survival. In this case, they were also able to identify antibody responses specific to these risk individuals that were harboring the genetic loss and received the transplant from an individual with a functioning copy. In the supplementary data, they also looked at long-term graft outcome, and they didn't find an association. We also checked the LIMS1 genotype in our cohort and also did not find an association with long-term graft outcome. So this LIMS1 single variant loss of function mismatch is associated with acute rejection, which does not transform into impact on long-term graft survival. We in Vienna, it's yet unpublished data, we also looked at the number of those loss of function variants, and we used a different approach. We again quantified the number of LOF variants, depending if you only look at the transmembrane protein, KIT-expressed proteins, or the combination of kidney and transmembrane as before. And we tested this variant with graft outcome. We could see the more of loss of function mismatches in transmembrane and kidney-expressed proteins you had, the worse your long-term graft survival was. So this is something we are working on currently, but this adds to this new concept that non-HLA proteins that are either polymorphic or absent are associated with an alloimmune response. So this is kind of the summary slide that includes this epitope concept in HLA and now transforms it to polymorphic residues on transmembrane proteins, and potentially include also these loss of function variants that share or that introduce a various number of epitopes that are mismatched or that are not present in the recipient and therefore are a potential driver for an alloimmune response. Thank you very much.